Yes, first of all, we have to remember that normally in normal cells, p53 is usually kept at a very low level to just ensure and mediate cell cycle arrest. And really, this low level of p53 in the cells is maintained mainly by MDM2. And indeed, MDM2 is the key negative regulator of p53 tumor suppressor protein. It modulates the activity by several mechanisms, as you could see. First, it inhibits, for example, the p53 transcriptional activity. It really transports it out of the nucleus, and MDM2 tags p53 for proteasomal degradation. This is normal. What happens in cancer? Next slide, please. A cancer cell is a stress cell with a lot of DNA damage. And what happens here, usually in many cancer cells, we have a dysregulation of p53 function, either because of the presence of an inactivating mutation in the TP53 gene, or we do have an overexpression of MD2. In both, by the way, just MDM2 stands for murine double minute 2, just to know for what this abbreviation stands. So either the overexpression or a TP53 gene mutation, they will overcome the p53-driven cell killing. This means that cancer cells get a survival advantage, and so cancer cells could proliferate. And that means that this brings us right away to the rationale for the use of MDM2 inhibitors to be used in cancer cells to try to restore normal p53 tumor suppressor function and thereby assist cells to go into cell death, the so-called apoptosis. Next slide, please. And indeed, one such example of an MDM2 inhibitor is the KRT232, or what is known as Navtemadlin. This is a potent, selective, and orally available inhibitor of MDM2. It has a very strong binding affinity. It can be rapidly absorbed, and it has a relatively long half-life of around 17 hours, which makes it also for the clinical practice and application a very relevant issue. Next slide. How does Navtemadlin work in cancer cells? As we said, Navtemadlin will increase levels of certain proteins that are very important for the p53 function, including p53, but others like p21, and this was one of the questions, by the way, MDM2, and as well PUMA. And PUMA is the pro-apoptotic BCL2 protein. And this happens in a dose-dependent manner. This is interesting to note that the compound does not only work on a protein level, but also on a messenger RNA level. For example, let's look at the p21 messenger RNA affection and increase under Navtemadlin in a dose and time-dependent manner. And as you all are aware, p21 is a really major target of p53 activity, as it links every cell damage, DNA cell damage, to cell death. So, taken together, the use of Navtemadlin will lead to cell death by inducing restoring the p53 function, which will induce the intrinsic mitochondrial apoptotic pathway and restores or stops the growth of tumor cells. Next slide, please. Well, the question now is, what is the role of MDM2 in myelofibrosis? As you all know, TP53 mutations are quite infrequent in patients with MPN, yet we know that the majority of patients, a distortion and a dysregulated p53 function is present. Why is that so? Today we know that through the JAK2-mediated signaling in the CD34 positive stem cells in patients with myelofibrosis, this leads to upregulation of the so-called La antigen. What is the La antigen? This La antigen can recognize specific classes of messenger RNA, including the MDM2 messenger RNA. So it will mediate the translation then the accumulation of the MDM2 protein. And so we see that in the majority of our patients with myelofibrosis, there is an overexpression of MDM2 in these CD34 positive cells. Next slide, please. So in summary, using the MDM2 inhibitor does have a rationale to restore p53 activity to drive apoptosis, as we have mentioned, by inducing the intrinsic apoptotic mitochondrial pathway, where the balance is shifted toward the pro-apoptotic BCL2 family members against the pro-survival apoptotic BCL2 members. Pro-apoptotic members, very important for cell death, like PUMA, again, it was one in the questions, NOXA, or again, BIM. And we know that the poor formers will lead then to the cell death by releasing cytochrome C and inducing caspase 9. So my next question is, okay, we have a rationale, we know that MDM2 is overexpressed in MF patients, but do we have a need for the use of this form of treatment in patients with MF, especially that we are now living in the JAK inhibition era? Next slide, please. The slides do not, yeah. So the answer to this is the next slide right away. The answer is, I think we all agree that the use of JAK inhibitors has completely changed the treatment landscape in many of our patients and improved their quality of life. Yet today we know that about half of our patients do discontinue ruxolitinib after 3 years of treatment, mainly not because of primary refractory disease, but because of loss of response or the so-called relapse. And then we have surely intolerance to the treatment. And we know very well that the prognosis of relapsed and refractory patients to JAK inhibition is really bad and their quality of life is bad. So there is definitely a need to have novel treatments in these patients after JAK inhibition failure. With this, I come to my summary of the first part of the talk. Next slide, please. So MDM2 is a key negative regulator of p53. We know that stem cells in patients with myelofibrosis have an overexpression of MDM2, and that if we inhibit MDM2 in these cells, we could restore the p53 tumor suppressor function and induce cell apoptosis. And we do have an MDM2 inhibitor called Navtemadlin, which has shown to be active in restoring p53 function. And we still do have an unmet medical need after failure of JAK inhibition in patients with myelofibrosis. With this, I will come to finalize this talk and jump right away to the second talk,